Good afternoon. Welcome to AP Biology, Proteins and the Nitrogen Cycle. So proteins and the nitrogen cycle, the cell needs both. And ultimately, first and foremost, it needs nitrogen, which it gains, where nitrogen comes from is through the nitrogen cycle. But we need nitrogen because it's in DNA. And DNA is what's used to code for RNA, which codes for protein. So you have to have both, and they're both really dependent on each other. So where do proteins come from? You've heard of proteins before. They're made of amino acids. You've probably heard of amino acids before. And DNA is what codes for the RNA, and the RNA codes for those amino acids in the proteins. So it all ultimately comes from DNA, which you can think of DNA as like a textbook. And that textbook tells the cell how to build all these different kinds of proteins. RNA is just a copy of DNA. So when we make a copy of DNA, we call that transcription, and the result is RNA. And RNA, as you can see, is single-stranded. It's just one long strand. DNA is double-stranded. And when we take that RNA, ribosomes use it to make protein, and we call that process translation. So how does translation work? Well, a ribosome, which is made of a large and small ribosomal subunit, takes that mRNA that was made during transcription, and it takes that RNA and reads it. And the RNA tells the ribosome how to build the actual protein. So these guys read the mRNA, which is like reading a textbook, a copy of a page in a textbook. And that page on the textbook tells this ribosome how to build a specific type of protein. And let's say that protein is a transport protein that's going to go to the cell membrane and transport sodium into the cell. We have free versus bound ribosomes. A free ribosome it produces proteins that are used by the cell, so this could be for metabolism of food. These ribosomes are freely floating in the cytoplasm. They're not connected to anything, so things that they make are going to be used in the cytoplasm of the cell, typically. We have bound ribosomes. They're bound to usually the rough endoplasmic reticulum, where they are producing proteins that can then be transported out of the cell. So they are for a specific function, such as digestive enzymes and polypeptide hormones. So you basically have two types of ribosomes, those that freely float in the cytoplasm, those that are connected to the rough endoplasmic reticulum, and the RER, or rough endoplasmic reticulum, is connected to the nucleus. So proteins, what are they composed of? They're made of amino acids. There are only 20 amino acids that actually code for functional proteins. So proteins usually have hundreds to thousands of amino acids in their structures. So proteins make up greater than 50% of an organism's biomass. The amino acid sequence is determined by the DNA sequence of the mRNA. So there's 20 amino acids, so in protein language, there are 20 letters in that protein alphabet, and they can build millions of different proteins with only having 20 different letters. Just like we can have millions of different words with only 26 letters in the alphabet. So there are 4 parts to the amino acids. They have a carboxyl end, an amine end, an alpha carbon end, and an R group. And we'll just look at this to show you. They have this carboxyl group, they have the amino group, and they have a hydrogen atom on the top, and then they have an R group on the bottom. The R group is what's going to change. That R group gives different amino acids their distinctive properties. Now that carboxyl end is going to act as an acid because it can release a hydrogen ion. So look at this carboxyl end here. It has a hydrogen that it can donate. So if it donates protons into solution, we say it is an acid. That amine group, this end can act as a base because it can accept hydrogen ions. So if you look at the amine group here, it can go from NH2 to NH3. So when you take 2 amino acids and you put them together, you get what's called a peptide bond. So the carboxyl end of one amino acid will mix with the amine group or the amino group of another amino acid. And you can see why they're called amino acids. One side's amino, one side is carboxyl group, which is an acid. So those 2 ends, those opposite ends, is what is attracted to each other, so it will connect and you get a peptide bond. So the monomers, mono meaning one, the singular units of a protein are the amino acids. The polymer, poly meaning many, the many units together would be your protein. So this is the peptide bond. If you'll look, you've got the OH on that carboxyl group and the H of the amine group, and they combine there to get your peptide bond. So what happens is that those 2 H's and that oxygen will combine to create a water molecule, and then what's left over reconnects, and you get your 2 amino acids connected. So there's different levels of these protein structures. If you look there at that primary structure, that's what would immediately be produced after a protein is made. So a ribosome, as we talked about earlier, it will put those amino acids, it'll put those amino acids together, yes, using the mRNA transcript. And it just looks like a string of beads. I like to use the analogy of a bracelet with many different charms on it. So you've got like a cubic one, a triangular charm, maybe a diamond, a star, all these different things put together. And each of those different shapes would be the amino acids. All of it together makes the bracelet. And of course, it wouldn't be circular, it'd just be a string of beads. So that'd be your primary structure. Now what you can do is that primary structure doesn't have a function. But if you take that primary structure and you fold it up into different ways, you can get structures that do have a function. So if you take that primary structure and you start to curl it around itself, you get a secondary structure, which you get all kinds of interesting bonds here between the edges of the amino acids, the individual amino acids. So you can spiral it. You can take that secondary structure and spiral it in many different ways. You see it's zooming out here, and all these different spirals are intertwined and connected. That's your tertiary structure. And then if you take many different tertiary structures and put them together, you get a quaternary structure, which is what we see right here, with 4 different proteins put together, 4 different tertiary structures put together to get one big quaternary structure. So primary structure is simply a sequence of amino acids. So where does the sequence of amino acids come from? Well, the coding for it ultimately comes from the DNA. The central dogma of biology is DNA to RNA to protein. Secondary structure, this refers to the shape of specific sections of each polypeptide chain. So secondary structure is after you folded it. You have alpha sheets or alpha helixes and beta sheets. So these are held together by H bonds during the cooling and folding. If there's no folding, there is no function. So a long string of beads, that primary structure, has no function. But with the alpha helixes and the beta sheets, then you start getting some function. So R group polarity determines how the protein will fold. We'll actually do a lab where you guys are going to fold proteins using like foam and these little Tic Tacs. And we'll show you what that looks like, or tacks, not Tic Tacs. So how are these proteins actually made by the ribosome? So the ribosome reads the mRNA. So remember, you've got your DNA, RNA, and protein. DNA and RNA are almost the same thing. RNA is just a copy of DNA, and it has uracil instead of thymine. So we take DNA, we make a copy of it, which is mRNA. The mRNA goes to a ribosome. The ribosome reads the information on the mRNA and makes protein from it. So mRNA template and tRNA are used by ribosome to make proteins. So the mRNA is fed into the ribosome right through here between the small and large subunit, and it has these 3 slots in the ribosome that bring in, or tRNA comes into these 3 slots, and on the back of these tRNA molecules you see they have amino acids attached. So it moves from left to right, and the tRNA comes in from right and exits to the left. So it's going 2 different directions. So your secondary structure is individual, the first set of folding. You have alpha helixes and beta sheets, and then the tertiary structure is when you take that secondary structure and you fold it even more. This refers to the overall shape of each individual polypeptide chain. You get disulfide bonds. These are super, super strong. So when you get to this tertiary structure, this protein is going to be held into that place for a really, really long time. If you look here, you got sulfur to sulfur bonds. Those are very, very strong. So for the quaternary structure, this is when you take, like in this case, hemoglobin molecule, you've got 4 different tertiary structures sandwiched together. And what's crazy about that is you've got 4 different things coded by different regions of DNA that produce different RNA that make 4 different proteins, and all of those proteins mix together to make a new functional protein. Individually, don't really do much, but when you put all these together, you get hemoglobin. And hemoglobin binds oxygen and CO2. So without hemoglobin, we couldn't survive. We would not be able to get oxygen to our lungs, and we wouldn't be able to get CO2 out of our muscles. So this is when 2 or more polypeptides are woven together to form the overall structure of a protein. So to summarize the 4 levels of protein structure, you got primary, sequence of amino acids. Secondary is interactions between adjacent amino acids. And then tertiary is that first set of 3D folding. And then quaternary is when you put many of those tertiary structures together. So primary is just that string of beads. Secondary is you spiral it, alpha helix or a beta sheet. Tertiary, you take that, you see it's zooming in here to what that part of the tertiary structure would be. So it spirals through here, but they take that spiral and they bend it and they bend it. Kind of like the supercoiling with DNA. And then you take these tertiary structures and sandwich them together to make a quaternary. So why is it so dangerous when you get a fever? Well, a fever is a sign that your body is getting really hot. It's overheating. And your body does this to increase its immune response to a bacterial infection or viral infection. Increasing that temperature actually helps your body fight off these infections. But if the infection is really bad, your body may get an out-of-control fever, and it will get so hot that it will actually destroy the proteins that are in your body. And if your proteins are destroyed, well, hemoglobin's a protein, or it's 4 tertiary proteins sandwiched together to make a quaternary protein. If you heat that up too much, then those proteins will denature. So if you get to about, I forget what it is, maybe it's 106, 107 degrees, proteins denature in the human body. So you've got your hemoglobin proteins, and it gets too hot, they'll denature back into the primary structure. And with no folding, there's no function. So no shape, no function. Shape equals function with proteins. So if you lose that shape, you lose the function. And if we're talking about hemoglobin, well, now because of your fever, you can't get oxygen to your brain, so you get brain damage. You can't get oxygen to your muscles and your heart, so your heart will stop beating, and you will die. So denaturation, here it is, temperature of about 106 degrees Fahrenheit. So if you've got a fever of 103, you're really getting close to having a fever that's going to kill you. So if you got this hot for a long period of time, you would definitely die. So temperature will denature proteins. pH changes, drastic pH changes will also denature proteins. If you put a cell in an acidic solution, if it's too acidic, it may not boil the cell, but just that change in pH will make the proteins denature, and then that's going to cause the cell to die because proteins are keeping it alive. Salt concentrations, if it gets extremely salty, that may cause proteins to denature, and denaturation disrupts secondary and tertiary levels of structure. So it may not be destroying the proteins, but if the proteins denature to their primary structure, they've lost their function, which they might as well be destroyed at that point. So a question for you would be, which one, hydrogen or sulfur or disulfide bonds, would break first? So H bonds or disulfide bonds? Well, we know H bonds hold water molecules together. They're strong, but they're not too hard to break. You can break them just by, heating water up, and it vaporizes, or it evaporates on its own. So H bonds aren't exactly extremely hard to break. Disulfide bonds are really, really strong. You have to heat them up to 106 degrees before they start to break down. So disulfide bonds would be the last thing to break. Hydrogen bonds would be the first. So we're going to talk briefly about the nitrogen cycle. Remember, nitrogen, the important thing about nitrogen is you need it for DNA. And nitrogen is in limited supply. And nitrogen molecules, they're N bonded to N, a triple bond. And as you remember, hopefully from chemistry, that distance between the nuclei is really small. You've got 6 shared electrons in there, so there's a lot of attraction to that space, that electron density between the atoms, so it's a really, really strong bond. So it's really, really hard to break. There's very few things on our planet that can even do this. And if we didn't have those organisms that can break this bond, we wouldn't have DNA, and we wouldn't have any living thing on Earth. So the nitrogen cycle is this process of how nitrogen cycles through our environment. And this is one of the big biogeochemical cycles you have to be familiar with. And really it's not much to memorize so much as being able to interpret it and why these things are important. So why is nitrogen important? Well, nitrogen bases make DNA and RNA. Adenine nitrogen base is using ATP. It makes amino part of amino acids. So you couldn't have proteins, you couldn't have ATP, and you couldn't have DNA or RNA without nitrogen. So it's extremely necessary for life. So how do we get nitrogen? Well, when it rains, nitrogen in the atmosphere, which nitrogen is in the atmosphere, it dissolves into the rain. You can dissolve a gas in water, right? Carbonated sodas, they have CO2 dissolved in Coke. So you can dissolve nitrogen in water as well. And when it rains, that rain is inevitably going to pick up a certain amount of dissolved nitrogen gas. Then that rain sits on the soil, and then it runs into the earth. And then it interacts with organisms that can take that nitrogen as a gas, that N2, and actually break it down and convert it into other things. And these organisms are bacteria. We call them nitrogen-fixing bacteria, and they convert this nitrogen into ammonium ions. And those ammonium ions can actually be absorbed by plants, and then those plants will use those ammonium ions. They'll break them down and use the nitrogen to make proteins, DNA, and RNA. So other ammonium is converted to nitrate by nitrifying bacteria. Other bacteria convert the nitrates into oxygen gas, nitrogen gas. This is called denitrification. So the nitrogen cycle, all the bacteria, the rain, the plants, the animals that eat the plants, the animals that die and decompose into ammonia, which is why they stink, they all play a role in the nitrogen cycle. I think I have a diagram here. A term you need to be familiar with is ammonification, converting ammonia into ammonium. So converting that ammonia ion into ammonium ion. We call that ammonification. So usually with AP Biology, they give you diagrams and ask you to explain. So you may know nothing about it and still be able to explain and get the points, but it helps if you've seen it before. So again, basically, the way we get the nitrogen out of the atmosphere in the first place is through rain. The rain absorbs the nitrogen gets in the water gets into the soil. Bacteria pick it up. You'll notice, like some of these roots on these plants, the plants have adapted to create these tiny little nodules on their roots. And these nodules are like little homes for bacteria. And these little homes that the plant creates are perfect environmental conditions for nitrogen-fixing bacteria. So it's a symbiotic relationship between the plant and this bacteria. The bacteria goes into these little nodules, these little homes built by the plant, which is the little root holes or nodules, and they grow in there and they absorb nitrogen from the water that came from the atmosphere, and they convert it into a form that can be absorbed by the plant, and the plant uses that nitrogen to produce proteins, DNA, RNA for its structures. And then we eat that fruit, and then we get nitrogen from the plant. And this can go through the soil, nitrogen-fixing soil bacteria, converted it to ammonium. Nitrifying bacteria can convert it to nitrates. Nitrifying bacteria to nitrates nitrites to nitrates. And then you can have denitrifying bacteria that convert it back into nitrogen gas, and it can go back into the atmosphere. And then you have decomposers that can also convert the dead plant tissue back into ammonium, where it can then be nitrified, and then denitrified. So it's this perfect little cycle of nitrogen going from the atmosphere and circulating through these organisms. And the way we get our nitrogen is ultimately from plants. And even if you're eating animals, those animals somewhere along the line are eating plants. And that's where all the nitrogen's coming from. So we'll go through a few AP questions here. So it says the figure above represents a rough endoplasmic reticulum. Which of the following best describes the role of the structure labeled Y? So this is a rough endoplasmic reticulum. It surrounds the nucleus. These little bumps on the RER are actually ribosomes. So let's see, which of the following best describes the role of structure labeled Y? Structure Y contributes to the raw materials required for the synthesis of proteins. No, that would probably be tRNA. Structure Y packages proteins for export from the cell. No, not necessarily. That would happen in the Golgi apparatus. Structure Y is the location where proteins are synthesized. That's definitely true. Let's see what D is. Structure Y contains enzymes that cut and activate proteins. Yes, so the answer would be C, because these are ribosomes. And ribosomes are where proteins are synthesized. Here's another question for you. Beaked whales feed at various depths, but they defecate at the ocean's surface. Nitrogen-rich whale feces deposited in surface waters supply nutrients for algae that are eaten by surface-dwelling fish. Which of the following best predicts what would happen if the whale population decreased? Okay, so these whales are feeding and they're defecating, and their defecation, their feces, is full of nitrogen. And the nitrogen is necessary for all the algae below these whales. So what would or which of the following best predicts what would happen if the whale population decreased? A says there would be a reduction in surface nitrogen concentration, which would cause an algal bloom. Well, if you're losing nitrogen, that wouldn't cause anything to bloom because they need the nitrogen. B says the surface fish populations would decline due to reduced populations of algae. That's kind of got the whole relationship backwards. No, the surface fish, not talking about the beaked whales. Potentially, let's see what our other answers are. C says the remaining whales would accumulate mutations at a faster rate. Not necessarily. D, the remaining whales would be forced to forage in the deepest parts of the ocean. So, if the whale population decreased, that wouldn't necessarily change where these whales would go, because essentially they're feeding on plankton. So they're not feeding on the algae. Why would less whales make them have to go to different places to find food? It also wouldn't affect their mutation rate because you have less whales. More whales, you have more opportunities for mutations. So there's no reason the mutation rate would be faster. So I think the answer would be B. Surface fish populations would decline due to reduced populations of algae. So, what would happen if a cell no longer had access to nitrogen? We talked about the nitrogen cycle. Well, nitrogen is necessary for DNA, RNA, amino acids, ATP. If you didn't have nitrogen, you couldn't make any of those things. You need ATP. This is adenosine triphosphate, ATP. You need that for energy. Here's a double helix of DNA. You need that to code for RNA, to code for protein. So you couldn't make proteins, you couldn't keep yourself alive, you couldn't pass on your heredity to offspring. So none of this stuff would work. You couldn't have amino acids. You couldn't make proteins. You gotta have nitrogen for all those things.